In previous lessons, we learned a little bit about consolidation theory and synaptic plasticity. In this lesson, we'll merge the two concepts to paint a picture of what might be going on in a brain as we experience an event and then later retrieve a memory for that event. So here's our brain. We have our hippocampus here. We have perceptual regions like visual cortex, somatosensory cortex, auditory cortex that will be processing episode number 1. So episode 1 will have certain visual features processed by visual cortex, auditory features, sound features processed by auditory cortex, and maybe there's a feel to the episode. Maybe you're petting a cat or a dog or something. So here's episode 1. And all of that perceptual information is fed down into the hippocampus. So the hippocampus is going to be integrating that information from the high-level perceptual areas. In a sense, activity then in the hippocampus is going to be representing the complex features in the context of episode number 1. Now, stimulus-induced activation of a subset of hippocampal cells creates a hippocampal memory trace for event number 1. So there are lots of cells in the hippocampus, but the idea is not all of them are being modified by episode 1. Just a relatively small subset of hippocampal cells will be responsible for making a memory trace for event 1. Now, how those cells get allocated to a memory we'll take up in another lesson. So schematically here, and this is just sort of symbolic or kind of an abstract thing, not meant to mean that there are 3 hippocampal cells connected in a triangular fashion here, but we're just going to let this little triangular picture here indicate that these hippocampal cells have undergone changes that are representing episode number 1. Now, what are those changes? Well, the idea is there would be changes in synapse strength, right, at specific hippocampal synapses among these hippocampal cells. And we learned that plasticity-related proteins or products affect recently active synapses to establish late LTP. You'll recall that early LTP involved the insertion of extra receptors, perhaps modifying those receptors. These changes account for early LTP, and late LTP seems to require protein synthesis and may involve structural changes to the synapse. So we want to imagine then that episode 1 has caused changes, synaptic changes among a certain set of hippocampal cells, and that's what it is to sort of encode episode number 1. Now, there are cortical changes going on as well, because after all, the cortex was processing the perceptual aspects of the episode, but the idea is that any synaptic changes up there in the cortex are weak at this time. Further consolidation processes will strengthen the cortical memory trace, and that's what we meant by consolidation, that there's a reorganization of the memory where the cortex will become more and more active for later retrieval. Now we're going to introduce a term called sparse coding here to refer to the fact that it is just a small subset of cells that are responsible for encoding a particular event. And as we'll see, the nature of the circuitry of the hippocampus also allows it to make separate representations, non-overlapping, distinct representations for separate events, like event 2. We'll see here in a second. So let's think about event number 2. Well, event number 2 will have some, some different visual characteristics and auditory and tactile characteristics. So you're going to get a different, set of perceptual cells active in these different perceptual regions. But nevertheless, they will converge on the hippocampus. And the idea is that a non-overlapping population of hippocampal neurons will be responsible for encoding event number 2 as distinct from event number 1. Even if 1 and 2 share some characteristics, the nature of the circuitry of the hippocampus is such that it's very good at generating different outputs for very similar inputs. So event 1 and 2 might be similar in structure or some of their perceptual characteristics, but the hippocampus is good at sort of assigning these 2 events to different populations of neurons so that we can keep these events separate later on if we were to retrieve them. So this system allows the hippocampus to make unique memory traces for ongoing events. And that process is called pattern separation, the ability of the hippocampus to take similar inputs and encode them with distinct populations of cells so we can make distinct memories. Now, what's the hippocampus doing here then? The idea is the hippocampal neurons are going to act as a pointer or an index to activate cortical representations of the experience that together represent the totality of the experience. So the changes happening down here for event 1 and event 2 down in the hippocampus, these hippocampal cells are going to be used to kind of make contact with the cortical representation of the memory. So there will be a dynamic interaction between the hippocampus and the cortex. And early on, after an event, the hippocampal cells are critical for retrieving the event because the cortical memory has not been strengthened yet by consolidation processes. So consolidation is going to reorganize the memory so the cortical cells become more involved in future retrievals. But initially the idea is, a lot of neuroscientists use this sort of analogy that the hippocampal cells are sort of like pointers or indexes to reactivate a memory or reactivate the cortical components of the memory for event number 1 or 2. Now, in this picture then, we do see that we've got 2 memories traces for event number 1 and event number 2. So at a later time, if a cue, in other words, a sound that was present during episode 1 or event 1, if that sound is heard again, it will selectively reactivate the hippocampal cells that were involved in making the memory trace for episode 1. That sound cue reactivating those hippocampal cells, which then are going to reinstate a cortical pattern of activity. So the cortical memory traces will get reactivated that were associated with episode 1. So this function of the hippocampus is sometimes called pattern completion. Here we have a set of hippocampal cells that were sort of making synaptic changes to make the memory trace for episode 1. And that episode had multiple sensory aspects to it. It had sounds and sights and context, etc. If only one of those components is re-experienced, it can reactivate the whole set, the whole memory trace in the hippocampus, which then can reactivate, act like a pointer or an index to reactivate the cortical components of that memory. Now notice then memory trace number 2 is not activated by a cue from memory number 1. Right? We had those non-overlapping sets of cells so that we can keep these memories distinct. And the cue that activated memory 1 or episode 1 was a different sound than was occurring during episode 2. Episode 2 could also be reactivated by a different cue. So let's summarize quickly the contribution that the hippocampus is making to memory processing. First of all, here we see some dentate gyrus cells and CA3 cells and CA1 cells, and there is a well-defined flow of information through the hippocampus. Now, the circuitry, the nature of that circuitry allows the hippocampus to do 2 important things for memory processing: pattern separation and pattern completion. The pattern separation is this idea that 2 similar episodes that might share a lot of characteristics can be assigned to differing populations of hippocampal neurons, so we can make distinct memories for different events. The pattern completion idea is that if only one cue from a past event is present, it can reactivate the entire memory trace in the hippocampus. And again, then the hippocampus is going to be in communication with the cortex to help reactivate cortical cells that were involved in the memory.